Thank you for giving me the opportunity to speak about my work today, and I'm having a great time listening to all things regulatory genomics. It's been a wonderful conference. So I'm Narayanan Raghupathy, and the Jackson Lab. And I'm going to be talking about my ongoing work on our effort to understand allele-specific gene expression using RNA-seq data in F1 mice. Our understanding of gene expression has come a long way since we were able to qualitatively measure gene expression using a Western blot in the 70s, and then we were, I think, the advent of microarray technologies in the early or late 90s kind of revolutionized our ability to measure gene expression on a genome-wide scale and be more quantitative about it. And more recently, the recent advances in next-gen sequencing technology have, I think, kind of propelled our ability to measure and understand gene expression to the next level. For example, we can not only measure gene expression level more quantitatively, but we can also identify novel transcripts, novel isoforms, and we can do many, many things more. The biggest advantage, or one of the biggest advantages of sequencing mRNA using RNA-seq technology is that we can study variation in mRNA sequences. For example, just like actually any other part of the genome, genes can have sequence variations like simple single nucleotide polymorphisms or insertion or deletions. So let's say what happens if a gene has a SNP. I mean, this I'm showing here as a cartoon, the gene and a SNP that has two alleles, A and G. So basically, the SNP on the gene can lead to two allelic homologs, which I've represented here, basically one with allele A and the other with allele G. So what happens if this gene is getting expressed? Typically, we would expect that both the alleles or the allelic homologs with the allele A and G would be equally expressed, just like here. But for some genes, one allele may be preferentially expressed over the other. For example, here in this gene, the allelic homolog with the T allele is almost 90% expressed compared to the allelic homolog with the C allele. So this preferential expression of one allele over the other is called basically allele-specific expression. Until now, although it's a really very interesting phenotype, it has been really difficult to study this on a genome-wide scale in an unbiased manner till now because of the lack of technology. People have looked at on a more targeted sequencing approach before, but for the first time, I think we are able to study on a genome-wide scale in an unbiased manner using RNA-seq data combined with good experimental design in a model organism. So this allele-specific expression basically can be due to both local genetic effect or due to parent of origin effect, where the parent of origin effect is, an example of that would be a complete imprinting, where the one allele is expressed depending on which sex of the parent it originated from. So I think we've kind of slowly beginning to understand what allele-specific expression is and what are the role of local genetic or the parent of origin effect using next-gen sequencing, especially RNA-seq data now. So what we have been doing is that we are looking at allele-specific expression in an F1 mouse from two divergent mice strains. One is NOD, which is our non-obese diabetic mice, which is one of the classical inbred mice. The other is PWK, which is a wild-derived inbred strain. And these two mice strains are very highly divergent. I mean, they have at least 16 million SNPs between them. And actually, I think more than 90% of the genes have SNPs in them. So it's giving us a great opportunity to look at allele-specific expression by doing F1 cross. So what we basically do is that we take the NOD females and cross it with the PWK males to get F1 NOD PWK males. So this is basically one direction of cross, where then here in the F1 males, NOD is from mother and the PWK is from the father. And reversing the direction of the cross, what we do is we cross PWK females with the NOD males to get again F1 male animals, but this time PWK NOD male animals. So the advantage of using reciprocal cross is that we can not only identify allele-specific expression, but we can also differentiate whether the allele-specific expression is due to local genetic effect or due to parent of origin effect, I think, which will become more clear in the later part of my talk. This is a more complex and complete experimental design that we used. For each direction of cross, basically we have 2 age group animals, and in each age group animals we have basically we put them under 2 dietary conditions, one controlled diet, other is any experimental diet, and in each group we have 6 biological replicates. So overall you can see that it's a very well balanced experimental design with the 3 major effects: direction of cross, age, and diet. So we can ask more detailed questions on allele specific expression, not only on the direction of cross but also on diet and hopefully age as well. So what we did is that we basically extracted mRNA sequence from liver samples from these 48 animals, and then sequenced them in technical replicates using Illumina GA2X technology. So for the most of my talk, I'm just going to be focusing only on the direction of cross, that is the F1 mice from NOD PWK and PWK NOD. But I think hopefully I'll have a chance to talk about a little bit about the diet effect maybe later. So one of the biggest challenges in looking at allele-specific expression is quantifying allele-specific expression. A naive approach to quantify allele-specific expression would be to take a common reference genome, for example, here mice, black 6, and then align our F1 hybrid RNA secretes to them, and look at the known SNP, and then see how many alleles are from NOD or how many alleles are from PWK, and then get an allele frequency count. But the problem with this approach, actually you do this for all known SNPs across the whole genome and get a whole genome-wide allele-specific expression quantification. But the biggest problem with this approach is that it is highly, actually its biggest problem is the alignment bias. Since the common reference genome available is black 6, and we are looking at F1 hybrid mice from NOD and PWK, so if you look at the phylogenetic relationship, basically NOD is another inbred strain like B6 or black 6, which is very close to each other, while the PWK is a wild-derived inbred strain, which is very far from both NOD and PWK. So because of the high, I think the phylogenetic relationship and the high divergence among the strains, our alignment will be really bad. For example, even though this genomic location, there is no allele-specific expression, just because NOD is closer to black 6, NOD might align better, while the PWK reads may not align to this location at all. So giving rise to a chance that it looks like it's a more NOD-specific expression at this location. So instead, what we did is that we will basically overcome this alignment bias by using 2 genomes, one for the NOD, other for the PWK. So we basically used Sanger low coverage genome sequence data to construct a pseudo NOD genome and a pseudo PWK genome. And for each RNA-seq sample, we align to both the genomes so that we can look at the alignment features of the same read in 2 different alignments and then make some inference on where the read came from. So the basic idea is that if a read is aligned to both NOD and PWK genome, we can look at how well it is aligned. For example, there are basically 3 possible cases. One where this read align equally well to both NOD and PWK genome. So this is kind of non-informative read, which are not really, won't be useful. But another case could be the read aligned really well to the NOD genome, but it has at least, let's say, 2 mismatches, aligns poorly to the PWK genome. So in this case, we will call that this read is probably from the NOD genome rather than the PWK genome. Similarly, you can also see the other case where the read aligns well to the PWK genome but not to the NOD genome. So using this kind of approach, we can basically call, or we can get NOD specific or PWK specific alignments and use that as a count for our allele specific expression quantification. So once we have done that, we can look at all the genomic features, be it exons or gene level, and since we have NOD level and PWK specific counts, we can compute either NOD or PWK allele frequency using from the count data. So the simplest thing one can do is that just plot the histogram of the PWK or the allele frequency. So here I have plotted the histogram of NOD PWK F1 animals. So the x-axis is the PWK allele frequency, as it goes from 0 to 1, and reassuringly it is nicely symmetric. In the case of the first approach, which I said, we could see that really NOD specific expression really high. So while the 0 here is the NOD specific expression, that is NOD allele is preferred, while towards 1 is where the PWK allele is preferred. So you can see that the allele specific expression is prevalent. Sorry, I probably missed this. So this is the, around 0.5 is the region where majority of the genes which do not show any allele specific expression. So from this histogram, you can see that allele specific expression is prevalent, and also it's more quantitative in nature. It's not just NOD or PWK, but you also see there are somewhere in between them. But since we're interested in identifying whether the allele specific expression is due to local genetic effect or a parent of origin effect, for that what we can do is we can compare the allele frequency from one direction of cross. That's what I have here on the x-axis, NOD PWK F1 allele frequency, and then compare it with basically the other direction of cross here. That's what I have here, where PWK NOD F1 allele frequency. So you see a strong diagonal which kind of suggests that both the allele frequency quantification kind of agrees, and in the middle you see the genes that do not show any allele specific expression, and the extreme corners of the diagonal, for example here, these are the genes that show NOD specific allele specific expression, and it agrees very well with both the direction of cross. But what is more interesting is that you also have kind of anti-diagonal elements, just very few of them, which show parent of origin effect. For example, let's take this gene, PEG3. What it shows is that this gene has a PWK allele frequency close to 1 in the NOD PWK F1 mice, suggesting that this PWK allele is preferentially expressed for this gene in the NOD PWK F1 mice. But if you look at for the same gene in the PWK NOD F1 mice, you can see that the PWK allele frequency is 0, suggesting that this is NOD allele is preferentially expressed. So you can compare these two and then see this gene's allele is preferentially expressed only when it comes from its dad. I mean, basically, it shows a parent of origin effect, and the gene's name kind of exactly reflects that. It is basically paternally expressed genes. So it is completely imprinted if it came from its mother. And similarly, you could see this, all the genes that are here, actually only two of them are here, are paternally imprinted. But on the other hand, if you look at the PWK allele specific expression that are along the diagonal, you would see that irrespective of what sex it came from, it is always expressed from PWK. And similarly, you would see that here it's always expressed from it came from NOD. So this suggests that this is a basically strong parent of origin effect here that is mainly from paternal expression on this part of the anti-diagonal, which we have IGF2R and H13, which are basically maternally expressed or maternally imprinted genes. And the striking aspect of this plot is that there are a lot of allele-specific expression that have local genetic effect, but only very few that show strong parent of origin effect. And all of these knowns are very well studied, I think at least even 10 years before, and this is not anything new here. And probably there are few genes that show not probably complete parent of origin effect, but partial parent of origin effect here. But we have, I mean, we can, I have the basically only a very handful of genes are there. And one thing that I missed here is that there are few genes that show it looks like it's parent of origin effect, but normally it's due to alignment artifact, which I haven't had a chance to discuss. So these are the genes that are from the X chromosome, but have, sorry, these are the genes that are from the autosomal regions, but have a similar gene in the X or mitochondrial. So it looks like it's maternally expressed while it just had alignment artifact. And our experimental design kind of can take care of that, because all the F1 mice are from all males. So you can look at X chromosome and then see what allele specific expression you see. Since, for example, if you look at NOD and PWK, since NOD is a mother, if you look at X chromosome, all of the genes should show NOD specific expression, but not PWK specific expression. But you do see some few genes suggesting that these are the false positive. I mean, it's less than 5%. So in that sense that you can expect, just due to the alignment artifact, about 5% of our findings could be wrong. And in the interest of time, I'll skip the current work and just to summarize. So we looked at allele-specific expression on a genome-wide scale using RNA-seq data. What we find is that allele-specific expression is prevalent across the genome, but the majority of the allele-specific expression seems to be influenced by the local genotype rather than a parent of origin effect. And finally, I would like to thank my postdoc advisor, Gary Churchill, and Ron Castagna, who did the vitamin D experiment, which I haven't had a chance to talk, and Fernando from UNC Chapel Hill, who are also part of the study, and the rest of the lab members. Thank you. I'll be happy to take any questions.
So we have time maybe for one very quick question. Okay. If not, thanks again. And